little bit about the etiology to begin with. So the virus is in the family Flaviviridae. It's another smallish RNA virus. The genus is pestivirus, and so that includes also bovine viral diarrhea virus of cattle and border disease of sheep. There's only one serotype of classical swine fever. You can divide it up into a number of genotypes and subgenotypes, which is quite useful for tracing purposes and seeing where the virus is coming from, but it doesn't really have much bearing on the disease. The virus has generally low environmental persistence, so it's a fairly fragile virus. It's an envelope virus, so you're looking at days to weeks, days in hot climates, probably like in the Caribbean. However, it is quite stable at low pHs, and what that means is that it survives very well in meat. So the process of rigor mortis, which for instance results in inactivation of foot and mouth disease virus, won't get rid of classical swine fever virus, and the virus survives exceptionally well. So in chilled meat, in frozen meat, even in cured meat products, the virus is still there and can readily infect pigs by ingestion. So this is an overview of the geographic distribution. Historically, a disease really of Asia and Europe. The situation in Europe has been much improved in recent years. There were problems with infection in wild boar, but that situation has been much improved. Still a lot of problems in Asia, and of course, we still have these problems in South America and in parts of the Caribbean. So this is just some figures that I could obtain on classical swine fever incidence from the OIE website. And I mean, happily there are a number of countries that have never reported or not reported since 2005, I should say, classical swine fever. But there are a number that have had classical swine fever in the last 10 years, and some of them you can see in red have very recent cases. So a little bit about the pathogenesis and the virus life cycle. The virus affects domestic pigs and wild boar. Those are the principal hosts. It's important to understand that it has a very variable clinical picture. So you have a wide range of virulence of strains, from very mild strains which are very difficult to recognize to the classical strains which produce the very obvious lesions which are more readily diagnosed. And for if you like, your average classical swine fever, which is moderate virulence, it's also important to recognize that there's age-related susceptibility, and you will see the full signs much more readily in young animals than you will in adults. So the virus enters usually through the mouth, and the tonsil is the primary target. The tonsil is very important. It's very important you know how to find a tonsil in a pig if you want to diagnose classical swine fever, because the virus is there for a long time. It starts there, and it's there all the way through the infectious process, so it's a useful organ to be able to get classical swine fever out of. However, you can infect pigs by other routes, so for instance by insemination, with contaminated semen, contaminated needles. If you don't change needles between injecting pigs, that's a very good way of spreading virus, because again the virus is in the blood for a long time. And then, like other pestiviruses, the virus is very efficient at crossing the placenta, so you have a range of effects of the virus due to that. Incubation period about 2 to 14 days. Virus shedding, again, as with so many viral diseases, can precede clinical signs, and it continues until death, and the virus is present in pretty well all the excretions and secretions of the pig. So if we look briefly at the pathology, when you grow classical swine fever virus in vitro, it's a non-cytopathic virus. It doesn't appear to do any harm to cells at all, and yet it causes this severe disease. And it's mostly due to the immunopathology and the fact that it is strongly immunosuppressive, and the body, in trying to overcome this, causes quite a lot of damage. So you get a profound leukopenia, you get a thrombocytopenia. Mention the tonsils, tonsillitis, you see. Other lymphoid tissues are affected, swollen lymph nodes, lymph nodes and splenic infarcts. As well as hemorrhage, you see hyperemia of the skin, and you get hemorrhages really throughout the body. Some of the common sites I've put down here. And then in the chronic forms of the disease, you get damage to the lymphoid tissue in the large intestine, and that can manifest as button ulcers in the cecum and colon. Okay, so the clinical signs that you more or less always see are depression, a very high fever, and anorexia. But the other signs you may or may not see, depending upon the virulence of the strains. So with mild strains of classical swine fever, rather non-specific signs are difficult differential diagnosis. With the classical strains, you get conjunctivitis, which is quite a useful sign. It's not that common in pigs. You often have constipation, which changes to diarrhea as the disease progresses. You can see nervous signs in the terminal stages, and of course you get this hyperemia and hemorrhage of the skin. Chronic strains, perhaps less common, but with these viruses you get undulant form of the disease with relapses, and it goes on for quite a long time, usually ends in death after about 3 months. So turning to prenatal infection then, as I mentioned, like the other pestiviruses, you get a very efficient transplacental transfer that can lead to fetal death, and then you can get either resorption of the pigs, mummification of pigs, and occasionally abortions. You can also get pigs that are born somewhat abnormal, so you can get congenital tremor as a result of classical swine fever infection, for instance, and you can also have stunted and poor doer pigs, and you can even have piglets that appear to be normal but develop a late onset disease which often exhibits as hemorrhages and looks just like classical swine fever. And from a point of other pestiviruses infecting pigs, this can be a problem for diagnosis, particularly for the laboratory diagnosis. But in fact, ruminant pestiviruses, when they infect pigs, don't really produce clinical signs unless you get transplacental infection. And then the pattern is very similar to congenital infection with classical swine fever. So I've got a few archive slides and also a number of slides from the 2000 UK outbreak. And you can have a look at the clinical signs we saw there. These pigs are on a fattening farm. They've got classical swine fever. You'd have a hell of a job to know that was classical swine fever, but they're huddling and depressed, basically. This is from an experimental infection, and it's the more classical signs. You can see the marked hemorrhages on the ear and some what looks like some rather unpleasant diarrhea going on. Again, classical signs of hemorrhages on the skin. This one, of course, looks rather similar to PDNS, which would be an important differential diagnosis. This is a sow from the UK outbreak. It's not hemorrhage, but it's what we call hyperemia and erythema of the skin, and the animal looks depressed. And you can also see a little bit of conjunctivitis there. And that's actually more marked in this picture. Again, another sow from the UK outbreaks. And this one shows the erythema very nicely on the udders. So this is the tonsil. It's nice and easy to see it when it's all inflamed like that, and you can see there are necrotic places on it as well. And here you can see with fluorescent antibody staining how the virus is in the epithelium and in the crypts of the tonsils. So these are some other lymphoid tissues that are affected, lymph nodes, which tend to be swollen and hemorrhagic. And this is the spleen with these infarcts around the edge of the spleen. This is the classical petechiation of the kidney, the so-called turkey egg kidney. And here you can see button ulcers in the large intestine, where the lymphoid tissue has been destroyed. So there's quite a large list of differentials for classical swine fever, especially when you start to take in the milder cases. But viewed as a hemorrhagic disease, of course, the important one is African swine fever, which Chris is going to talk about in a minute. There are a number of other viral differentials here, particularly circovirus infections, PMWS and PDNS, swine influenza, which we're going to hear about later, and PRRS as well. In addition to that, you'd have to think about a number of bacterial septicemias, certainly Salmonella erysipelas, and also various toxicological conditions that can lead to bleeding, such as warfarin poisoning. Okay, so routes of spread then, pig to pig horizontally, always the most efficient route. Pig to pig vertically, as I've mentioned, so sows can become infected, they can appear normal, and then they can give birth to piglets that are shedding a lot of virus later on, and via semen. Pig meat, we've mentioned again, very important if fed to pigs as swill. Of course, it's very readily inactivated, the virus, by heat and proper cooking. Over long distances, the big spread is lorries, and especially lorries that are not cleaned out properly between moving pigs. And indirect over short distances, we have various fomites and mechanisms for moving the virus around. And certainly in Europe, wild boar have been very important as a reservoir host. So the challenges then for early detection are the fact that it's quite difficult to diagnose and to recognize classical swine fever, especially if you don't get the most virulent and obvious forms of the disease. Again, some analysis looking at, in the Dutch outbreaks, what was the sensitivity of clinical diagnosis, and it was only around 50% at 37 days after virus introduction, went up to 90% by about 47 days. So it takes time before you get enough disease that you can recognize. So the low virulence of many virus strains that we've mentioned, the time that it takes before sufficient pigs become infected. Fevers aren't always that easy to measure, especially in large pig herds. If you visit tracings too early, you therefore will tend to miss the disease. And random virological testing, of course, is not really practical. And another problem for classical swine fever is that antibody development is quite slow. So if we're talking about foot and mouth disease, it's in about 5 days, you already get antibodies. Classical swine fever, immunosuppressive virus, you don't get antibodies for about 2 weeks. Okay, so then a little bit on the diagnosis, on the laboratory diagnosis. I've already mentioned that you get a lot of virus in infected animals in the blood, in the tonsils, in other lymphoid organs. So there are plenty of places you can get samples to detect virus, and really the best two are blood and tonsils. Nowadays, RT-PCR is the mainstay for diagnosis in most labs. You can do virus isolation, but it's quite hard work because the virus is non-cytopathic. You have all the problems of potential pestivirus contamination of your cell cultures or pestivirus antibodies present in fetal calf serum. So PCR is a good test. There is also an antigen ELISA, which is quite good, much less sensitive, but because there's a lot of virus in blood, you can also use that, and that could be useful to give you a backup and confidence in your PCR results. There are also, I haven't really come on to vaccines yet, but there is also a DIVA PCR, which can distinguish between the live vaccine and the field virus. So serology, as I say, the issue with serology is antibodies appear quite late, but they're very long-lasting thereafter. There are a number of different ELISAs that you can use to different viral proteins. So the major envelope protein, the E2 protein, you get a strong antibody response to that, and you can certainly measure that. Some of these tests can differentiate antibodies induced by vaccines and ruminant pestiviruses. VNT is the gold standard in order to do comparative serology. Genetic typing, you can use that to help you determine the source of your outbreak, where the virus has come from. So a little bit on vaccines. As far as recovery from classical swine fever, there's an important cell-mediated and humoral immunity, and either can protect the pig. So there are some vaccines that only induce cell-mediated immunity, some that only induce antibody. Both are actually protective. The most widely used vaccine is the modified live vaccine, such as the famous C strain vaccine, which has been used very extensively around the world. It's extremely effective. It works very quickly, very potent. And because it's a live vaccine, you can use it in oral baits. So if you're trying to control the disease in wild boar, that's very useful. Marker versions of this have been developed by recombinant engineering technology, but they're not, I think, licensed and available yet. Alternatively, there is an E2 recombinant protein, so this is the major envelope protein. It induces a good antibody response, and you can get quite good protection with it. It's probably not as efficacious as the live vaccine, and it takes a bit longer to develop protection, but you have then a good DIVA capability. A whole string of novel vaccines have been worked on. Viral vectors expressing mostly the E2 protein. They all are protective, but none of them are licensed. Similarly, there's a lot of work being done in Europe to look at various modified genomes wherein some of the virulence genes have been removed or swaps have been made with other pestivirus genes, but again, they're not licensed at the moment. So the principles for control then, in the EU, the policy is stamping out, so it's the same old things that I said about foot and mouth disease. You've got to detect the cases early. You've got to put movement restrictions on. You can slaughter the affected herds, do your tracings, possibly think about preemptive slaughter if you can't control the disease without it, and similarly emergency vaccination if it looks like the disease is going to get out of control. This is a tracing from the UK index case in 2000. So this was actually the first herd to get infected. We've got a whole breeding pyramid here represented. So you've got a number of breeder farms here. This is a large producer of fattening pigs. It then sold these pigs to a whole series of other farms where the pigs were fattened. So the disease was first recognized on one of these farms, and it wasn't actually recognized until they had a very high mortality because the vets thought it was PDNS. Okay, so the challenges for classical swine fever control, at least in Europe, is high density pig keeping, animal movements, infection of wild boar, globalized trading, so bringing meat. In the case of the UK 2000 outbreak, it was probably meat coming from Asia, and swill and scrap feeding.